Hello students, once again welcome to EPG Pathsala. I am Samar Singh from Department of Microbial Biotechnology, Punjab University. Today we are going to discuss about a module named Classification of Restriction Enzymes or REAses under paper Genetic Engineering and Recombinant DNA Technology. By the end of this module, you will have learned what is a restriction enzyme, restriction enzymes, discovery, their biological significance, naming convention for restriction enzymes, major types of restriction enzymes and their defining characteristics, miscellaneous restriction enzymes which are generally not included as a part of general classification. Restriction enzyme or endonucleases or REases are those enzymes that can restrict or make cut in a DNA molecule at an internal phosphodiester bonds, generating new free 5' phosphate and a 3' hydroxyl groups. As shown in the cartoon, a restriction endonuclease can cleave a DNA fragment or a polynucleotide duplex at an internal position, thus generating two fragments each with a new set of free 5' phosphate and a 3' hydroxyl group. The indication about the existence of these enzymes that would be responsible for cleaving the foreign invading DNA and the cognate modification or protection system that would be protecting the host cell's own genome from such restriction systems came from the bacteriophage plating experiments done in 1960s as illustrated in the next slide. Bacteriophage plating experiments are generally performed to assess the susceptibility of bacterial strains to bacteriophages, mostly DNA viruses that kill or lyse the bacteria. In the experiment, bacteriophages are plated on a lawn of cells of the test bacterial strain. If bacteriophages are able to infect the bacteria, they produce a clearance zone or plaques as a result of lysis of the cells. In the bacteriophage plating experiments, it was observed that somehow the ability of bacteriophages produced from one strain of bacteria remains quite high for the same strain of cells, but not for the other strain of bacteria from same species such as E. coli. Let us try to understand the statement made through the shown illustration. Suppose we have a bacteriophage preparation X that we use to infect two strains of E. coli, namely B and K. In the first plating experiment, we end up seeing only a few plaques in both B and K strain plates. But when the bacteriophages coming from B strain plate are used to infect B strain cells or alternatively that from K to K strain cells, the ability of bacteriophages to infect increases tremendously and remains so for the same strain, but remains always diminished for the other strain in the first round of infection. This observation led to the proposal that host cells must have a restriction system that restricts any foreign looking DNA, but its cognate modification system keeps the host genome modified so that the restriction system would not restrict it. Together, these two systems are referred as restriction modification systems. Restriction enzymes or restriction endonucleases. Werner Arbour in the early 1960s first discovered or proposed that their biological function is probably to recognize and cleave or restrict foreign DNA. As in the host cell, the DNA sequence that would be recognized by its own restriction endonuclease is protected from restriction or digestion by methylation of the DNA, catalyzed by a specific DNA enzyme or DNA methylase, which is also abbreviated as MTAs. The restriction endonuclease and the corresponding methylase are sometimes referred to as restriction modification system. They are found in a wide range of species. Prevalence of RM systems or REases. Restriction enzymes or restriction modification systems are quite well distributed among the species, but non-uniformly, and they may range from none to too many in different species. For example, in diverse species, at least in 90% of the genomes, at least one RM system has been found, but about 80% are supposed or have been shown to contain multiple RM systems. These restriction modification systems and hence REases are diverse and ubiquitous in prokaryotes. So far, about 4,300 enzymes with more than 300 different specificities are characterized or have been characterized. Recognition sequence of these REases. The recognition site of most of the REases is palindromic or partially palindromic. What we mean by palindrome? It is usually a word, phrase, or sentence that can be spelled identically when read either forward or backwards, such as Malayalam, Samas, Rotator, Mom, Dad. Palindrome or palindromic is defined as something having a dyad symmetry around an axis. Actually, what is a palindromic dyad or twofold symmetry? Dear students, let's see what constitutes a palindromic dyad symmetry or a palindrome sequence in case of a DNA molecule. In case of DNA, as you know, it's a double-stranded. So for being a palindromic sequence, it should have a region with inverted repeats of the base sequences having twofold symmetry over the two anti-parallel DNA strands, as depicted in the cartoon shown for the recognition site of BAM-H1 restriction endonuclease. Essentially, what it means is that to superimpose one repeat sequence over the other inverted repeat, such as the sequence shown in light green color or light orange, either one must be rotated first 180 degrees about the horizontal axis, then again rotated 180 degrees about the vertical axis, as indicated by the curved arrows in the figure. I suppose now you would be able to easily recognize a palindrome in a DNA molecule. Naming of restriction endonucleases. Smith and Nathans in 1973 first proposed and suggested the naming guidelines for REases. They suggested that a REa's name should begin with a three-letter acronym in which the first letter should be the first letter of the genus name and the next two letters should be the first two letters of the species name from which the enzyme was isolated. Additional letters or numbers could be added to indicate individual strains or serotypes, followed by a space. Then the chronology of identification of the enzyme is represented in Roman numerals. The first three letters of the name should be always italicized. For example, Hind3 is used for the third enzyme discovered from Haemophilus influenzae serotype D. Similarly, the name BAM-H1 is the first restriction endonuclease characterized from Bacillus amyloliquefaciens strain H. However, new guidelines for naming the REases have been proposed to accommodate the developments in the field in year 2003. Some prominent changes include avoiding the italicization of the three-letter acronym, removing the space between organism designation and Roman numerals. For more detailed nomenclature guidelines, please refer to Nucleic Acid Research paper that was published in 2003, volume 31, page number 1805 to 1812. So, REases which were previously named Hind space Roman numeral 3 should be written as Hind without space Roman numeral 3 and no italicization of first three letters. Now let's talk about restriction endonucleases classification. The restriction endonucleases are generally classified into four major types, namely type 1, type 2, type 3, and type 4, primarily based upon four differential characteristics with reference to which are: first, subunit composition of the enzyme; second, the cofactors required for the enzyme activity; third, the sequence recognition specificity of the enzyme; and the fourth one, the position of cleavage or restriction site to the DNA with respect to the recognized sequence. Classification of restriction endonucleases. They are generally classified into four different types, as you see in the table shown, based upon the subunit composition, cofactor requirement, recognition sequence site, and restriction site. For example, type 1, they are usually composed of subunits of R, M, and S as heterooligomers, while in case of type 2, you usually find homo, di, or tetramers of R subunit, while in case of type 3, we usually see heterotrimers or heterotetramers of the composition M2R1 or M2R2 respectively, while in case of type 4 subunits, so far we have seen single subunit that has both R and M activities. Here I would like to clarify: R means restriction activity and M means modification activity. They also, all of them differ based upon their cofactor requirements. For example, type 1 requires magnesium ions, ATP, as well as S-adenosyl L-methionine or adometh, while type 2 just requires magnesium ion for its activity. Type 3 again requires magnesium ion plus ATP, and type 4 requires magnesium ion for activity, but its activity is stimulated by the presence of adometh. And if we look at recognition sequence, as you can see, the type 2 generally recognize 4 to 6 base pair long palindromes. Type 3 require or recognize short asymmetric sequences of 5 to 6 base pair length, and type 4 usually require or recognize asymmetric recognition sequences that must have modified bases, usually methylated bases. And if we look at the restriction site or where the enzyme cleaves, type 1 usually cleave the DNA about 1000 base pairs away from their recognition site. Some examples of type 1 are ECO K1 and ECO R1241. In case of type 2, the restriction site is usually at or near the recognition sequence. So they usually cut within the recognition sequence. And in case of type 3 REases, they usually restrict or cut the DNA about 25 to 27 base pairs away from the recognition site, which is usually two recognition sites in an inverse orientation. The examples of type 3 enzymes are EcoP1 and EcoP151. Type 4 enzymes generally cleave the DNA at about 30 base pair or more away from the recognition sequence. One example of type 4 is EcoKMCRBC enzyme. It should be noted that so far the enzymes that have been characterized, about 98% of the total enzymes are type 2, while in the 2% you have type 1, type 3 and type 4. This is the visual depiction of the classification of enzymes that we have just discussed, and they show how different subunits are together in type 1, type 2, type 2s, type 3 and 4. And this is adapted from restriction modification systems by Robert M. Blumenthal and Xiao Dong Cheng in Modern Microbial Genetics, second edition, that appeared in year 2002. Subtypes of type 2 restriction endonucleases. As we just discussed that about 98% of the restriction endonucleases are type 2 restriction endonucleases. They have been further divided into various subtypes based on unique properties as shown in the table below. But remember, some REases may belong to more than one subtype. Subtype A, they usually recognize asymmetric sequences or their recognition sequence is asymmetric. One example is FOK1. Then subtype B, it cleaves both sides of the target on both strands. It's unusual in that sense. While most of the other REases cut at single side, this class 2B cleaves at two sides on both strands. Example is BCG1. Subtype C, they recognize symmetric or asymmetric target, and the restriction and modification function is in one polypeptide chain. The subtype E, they usually recognize two targets when both of them are together, and they cleave one target while the another target acts as an effector. Subtype F, they recognize two targets and both targets are cleaved coordinately. Type 2G, they also recognize symmetric or asymmetric targets, but their activity is affected by the presence of adometh. Type 2H, they recognize symmetric or asymmetric target, and their overall subunit structure is similar to type 1. Type 2M, this is subtype of 2P or 2A, and they require methylated target such as DPN1. Remember, most of the DNA cutting these restriction endonucleases recognize and cut unmethylated substrates, but this DPN1 from this that belongs to group 2M or the restriction endonucleases that belong to type 4, they can cleave methylated targets. Subtype 2P, they recognize symmetric target and cleavage sites. Type 2S, they recognize asymmetric target and cleave it. Type 2T, they recognize both symmetric as well as asymmetric targets, and their restriction genes usually make heterodimers. Example is BPU101 or BSL1. This slide again gives you example of some commonly used restriction endonucleases, their recognition site, and the information about their cleavage site. For the first example is BAMH1, which recognition sequence is CGA-TCC, and it cleaves within that sequence in between the two terminal Gs. Similarly, there is a CLA1 enzyme that also recognizes sequence ATC-GAT, and it also cleaves within the sequence. Other examples are EcoR1, EcoR5, Hey3. Here, the position of cleavage is indicated by arrows, as you see, and wherever there is a star, it denotes the bases which, whenever modified or get methylated by methylases, they prevent the cleavage from the REases. Miscellaneous REases, these are restriction nucleases that we seldom talk about. First, let us talk about homing endonucleases. They are a special class of endonucleases found in eukaryotes. They are encoded by many group 1 and group 2 introns, which are also mobile genetic elements and supposedly parasitic genetic elements. They promote the movement of introns to new identical sites in another DNA copy of a homologous gene that does not contain the said intron. In a process termed homing, whenever the HEAs encoding DNA intron is introduced into a cell by a parasite or by other means. They usually recognize and cleave much longer DNA sequences, which are usually more than 14 to up to 35 base pair long. Nicking enzymes. There are two kinds. First type, those that behave functionally like restriction endonucleases, but they just cleave one strand of the DNA substrate. For example, N.BSTSE1 enzyme. Its recognition restriction site is GAG-TCNNNN. It's also abbreviated as GAG-TC4. The arrow indicates the position of restriction made in the single strand. Note here the prefix N denotes its nicking activity to differentiate it from common REase activity. Second type of nicking enzymes, they are generally found in association with enzyme M-05C methyltransferases, and they make nick adjacent to G-T mismatches, which generally result from deamination of methylated cytosine within the recognition sequence of the specific methyl transferases, such as VSR protein that accompanies the DCM methyltransferase of E. coli K12. It recognizes the specific G and T mismatches that occur if there is deamination of the methylated cytosine residue within the context of CCWGG recognition sequence of the specific methylase. Students, now let's summarize the module we have learned so far. Restriction endonucleases are almost ubiquitous endonucleases that show sequence specificity to recognition site and or the cleavage site, and most of them are part of restriction modification system in a cell or in organisms that help protect the host genome from invading genomes. The methylation of recognition sequences, mostly palindromic, prevent restriction endonucleases activity. Exception is DPN1 that actually recognizes a methylated sequence and only then it cleaves it. There are four types of restriction endonucleases based upon the subunit composition, cofactor requirement, recognition site, and cleavage restriction site position. The type 2 restriction endonucleases are the most common restriction endonucleases that recognize 4 to 8 base pair sequences, and they only require magnesium ion for their activity, and they cut within or very near to the recognition sequences, whereas type 1, 3 and 4 have more stringent cofactor requirement and they cut the DNA away from the recognition sequences. Thank you.